afternoon, everyone. My name is Tanu Soni, and I'm going to talk a little bit more about lipidomics, data analysis that we do here in the core, and the basic report that we give to our clients. So lipidomics already described, and as we know, lipids are a part of, is a subset of metabolomics, and lipids are basically, they share common physical and chemical properties, and they can be divided into classes. That's how they are different from rest of the metabolites. And out of all the omics, lipids is a fairly new entry, and still recently, because of the advances in mass spectrometry based techniques, we have it has gained popularity, and lipid metabolism is, I would say, like least explored metabolism in terms of all metabolic pathways. And the reason is that we don't have all the pathways for lipids. So I'm going to talk about four things, important things in this talk. I'm going to talk about lipid classes that are covered by our platform, and then what is the workflow of our pipeline, the data processing and the data analysis steps, and then I'm going to show you one example report that what we give to our clients. So the lipid classes, mainly three big classes that are covered are glycerolipids, glycerophospholipids, and sphingolipids. Glycerolipids mostly elute in positive mode. Glycerophospholipids mostly elute in negative mode, but glycerophosphocholines and glycerophosphoethanolamines appear in positive mode as well. Sphingolipids we see in sphingomyelins we see in positive mode. Ceramide phosphates we see in negative mode, but we don't see that many. Ceramides, there's one more class which is ceramides. We see those mostly in positive modes. These are the common ones we see right now in terms of just the samples that we have identified. But if there is a particular class, and we can include that also in terms if you're interested in anything particular, and we've been working on these methods. So this plot shows a class-wise distribution of all the lipid classes. As you can see, TG are the most abundant in positive mode, followed by PCs, and PCs are the most abundant here, followed by phosphoethanolamines. So now let's diverge a little bit and talk about databases. And this question today also came up, and somebody was asking me that, and I'll explain the reason why we are talking about this. So lipid maps is the first internationally recognized classification system for lipids that we have. This is a great resource for all the lipid information that you can get. And it has more than 40,000 unique lipid structure, this lipid map structure database. You have some good tutorials about lipids. You have not exactly pathways, but information related to pathways and pathway creation tools. Then you have some MS-based search tools in here. And also there are like 500 standards, MSMS spectra for 500 standards that you can get from here. And even though lipid maps is a great tool, I would just like to point out that why the identifications from our report cannot be directly mapped to this lipid map structure database. So lipid maps is a great tool, and they have the basic hierarchy for the classification that we have right now are three main levels: the category, the main class, and the subclass. And this figure is from a paper from EDI database, which is called Lipid Home. And what they have done is they have reclassified these things a little bit and added 4 more classes here, and which makes it a little bit easier to understand why these can't be mapped here. So the reports that we give here at the core, we give information at the species level. We cannot quite give this information, exact information of what the fatty acid scan species are, because each of these, for example, PC36-2, which is 36 carbon and 2 double bonds, can have different combinations of the fatty acid chains. So once you have your report and once you have identified, uh, you know, some lipids that are of interest to you, and like Soup talked about it yesterday, you have to go for a validation by target analysis, and that's where we can, find out more information about these scan species and then confirm what these scan species are. So for the workflow, data processing steps, I'm going to discuss data processing steps in the pipeline, the quality control that we do, data normalization, briefly what we do here, and data analysis. So, the data processing workflow starts with the sample submission form. Sample submission form is filled by the client. They give all the information, factor information associated with these samples, and just a basic write-up about like what they're trying to do with this. And once we get this information, sample information is uploaded in our LIMS system. And keeping this information in mind, we take the experiment design into consideration and randomize these samples before we put it into mass spec for the run order. And tomorrow, I think George is going to talk more about these randomization and design. I think Anna has also touched a little bit about this thing, why it is important to do this kind of randomization before you run these samples. So once we randomize these run orders and put it into mass spec, and then we collect MSMS data on all the samples. Once we have raw data files, we search all these raw data files through Lipid Blast. Lipid Blast is a in silico MSMS spectra database. So we search all these files through Lipid Blast database, and we get the identification hit for each of these sample files. So let's say if you have 100 samples, we get 100 files separate for these 100 identification files for these. Then what we do is we combine all these 100 identification file and create a master library. And then from then on, we go to a little targeted search. We create this master list and we use the multi-quant software by AVSciX, which is for targeted analysis. And then we search for each compound identification based on mass and retention time, and search the peak areas, integrate the peak areas as our raw data. So this is just to show how Lipid Blast does the identification in terms of spectra. Below, you can see is the Lipid Blast spectra. That is, and on the top we have the spectra from our experimental data set. And as team explained, this is one of our standards that we spike in PC340. One characteristic fragment for choline is this peak 184, and this is obvious that this is going to be the highest peak. So this matches right here, and there are a couple of other peaks that match here. Based on how many peaks match, a score is assigned, not just based, but the characteristic peak and the other fragments, how they match, a score is assigned to every identification. And also, lipid maps give the fragment formula, or so to say, the fragment information, what each of these fragment could be, how they're coming from this total formula. So this gives a detailed information of and how many of these peak match can give you like probably what the identification is. This is another example, PE340, which is also our internal standard, and the characteristic neutral loss here is 141 neutral loss, which is this 579 peak. So this So the quantification, after the identification is based on the MSMS data, and quantification is done on MS1 data. This is again, TM showed a similar plot where we show how each of these classes elute, lysos elute early in the retention time, as DGs, PCs, and Ps come somewhere in the middle, and TGs and cholesterol esters come towards the end in the chromatogram. So for data analysis workflow, we start with the raw data peaks that we get, peak areas. This is our raw data. And then we look at, then we do the QC analysis. And then we go for normalization. And if any pre-treatment is required, we do data pre-treatment. And from then we go for univariate, multivariate analysis. QA, QC, so three important things we use here: internal standards. So internal standards, we usually have internal standards for all the classes. So what we do is we create a mix of all these internal standards and spike into each sample before running them. Another sample, QC samples, are these pooled plasma samples. These are the pools from Red Cross, and these pooled plasma samples we run with each and every experiment, irrespective of what the experiment is. Then the third type of QC samples are test pool samples, which is basically the aliquots from that experiment, small, from each sample, and a pool is created, and this we run like after every 10 or 15 runs to monitor the variations when we're running it. So this plot is for our internal standards, and when we check the internal standards for the variation, how the internal standards are behaving. If the all these internal, this is inter-day variation, and as you can see, all these internal standards here are pretty tight. All of them are below 9% coefficient of variation. And if any of the internal standards we see has high variation, we go back and check which sample has the variation, what is the issue, if it's like a missed injection or if it's just happening in specific regions, because these classes elute at a specific region. So this gives us an idea that what needs to be done. And also based on internal standard reproducibility, we select our internal standards that we use for normalization. So variation, other than two important types of variation that we have here, and analytical variation we see and we know, and sometimes people forget about this type of variation that can come in the data, like for biofluid type. Tissues that we get mostly are of different weights, so you need to normalize for that. And then urine samples a different concentration, and cell line, it's very hard to exactly measure the exact cell count. So for these variations, you need to normalize the data, especially for tissues. We normalize by weight, and for cell line with protein content, and urine mostly we use creatinine. And another variation is analytical variation that can come from sample preparation, or instrument run order, drifts due to batches, or and then for this type of variation, we use all our three types of controls that I discussed: the internal standards, and then the quality control samples. Once we take care of these variations, then this improves the downstream statistical analysis a lot, because we are able to separate the biological variations from this systemic variation. So I'm going to briefly tell the normalization method that we use regularly for lipidomics, but I'm going to talk a little in detail in the hands-on session about this. So for lipidomics, the relative response to internal standard works pretty well if we don't have any other kind of batch effects, just normal relative response to internal standards. We have so good, sometimes like data is so good that we don't even need to normalize it to internal standards, just any transformation like variance minimizing transformation just can work pretty well with the data, and you can just start the data analysis. But we use relative response to internal standard, class-wise or retention time based. If you don't have a specific standard for a specific class, you can use based on the retention time, or you can use based on the medians of all the internal standards, or you can also use it based on variance minimizing, like which one is minimizing the variance most. For when we run like a large number of samples, we do see batch effects. So for this, this smoothing technique, Lois, works pretty well with our data sets. And other than that, you can fit a regression model to adjust for batch effects. Though a little bit of caution here, it, we use our QC samples to train this, to train our span parameters so that we don't overfit our data. So this is just one example from the data report that we give to client, and this is the information that we collect. So this is an example data set, and these are, so in this data set we had kind of two separate experiments going on here. So these are NIH rats from selected based on their training response, or selected based on their running capacity. So these are two different experiments we are trying to do, and I'm going to call this experiment 1 and experiment 2, just to be clear. So in this experiment 1, we have the animals that are selected based on their low response to training and high response to training, N of 8 each. And then these animals are 2 years old, and they were trained for 8 weeks and improve their oxidative capacity. And what we are trying to identify here is if there is an effect of training in these animals in both LRT and HRT. And then the second experiment is these rats are selected based on the running capacity, and these are low capacity runners and high capacity runners. And we have 3 groups here. We have young animals, and then we call these in the middle are mature, and then these are old animals. And we're trying to identify if there is an effect of age and if this effect is different in different animals. So we start after we run after we run these samples. We first start at looking at the just the overall TICs to check for any unusual drifts in the retention time, or if anything else, samples drop out or any missed injections, to see if everything went fine. Then what we do is we just take all the data and just try to visualize everything unsupervised way and check how the pools look. The pools here are our test pools are in the center, and they're pretty good. We see slight drift here in one of the sample, but all of them are like pretty good here. That's what we expect to see. Our plasma, human plasma, is right here outside, and all these are rat plasma. So this is together. So another thing is that since we ran all these samples together, like the experiment 1, experiment 2 that I said, so I'm going to, we did normalize them all of these samples together. So, and then we check the internal standards in each mode, and most of the internal standards here are pretty good, pretty reproducible, and we have and all the rest of them are below 25%. Same is for the negative mode. It looks pretty good, and we use all the internal standards for normalization. And this is our plot after we've normalized the data. So it looks pretty good, very similar. We haven't changed or modified our data in any way. And this is just to show you along with the basic QC, we give a Word document of like describing the method and write up about the whole process, including including the sample prep and everything in the report. Plus, we give this Word document for all the data and the data analysis that we do. So this first tab is for the information, data information, because when we get these samples, we give the sample IDs internally to all these samples, and the group information and all the other information that is submitted by client is mapped to this. And then we give the raw data for each mode, both positive and negative mode. So the raw data, most and all of our lipids are identified uh, because we are using kind of a semi-targeted search. So the name of the lipids, and then we give the RSDs for our test pools. Then the class information, what class they belong to, then the mass window where the compound is searched on, and the retention time. And this tag here that we add is known unknown is based on if the lipid has an odd chain fatty acid, then we call it unknown, because odd chain fatty acid are not that common. So we call it unknown, and rest of the even chains are named as known. Then we give for both each mode, we give separate normalized data spreadsheets, and this just has the names and the normalized. Then this is what we've recently added. What we do give also is a combined report, a combined version of both negative and positive normalized data, because some of our classes appear in both positive and negative modes, especially PC and PE. So the way we combine this is, uh, so if a compound appears both in negative and positive, let's say some compound PC36-2, if it appears both in negative and positive mode, then we select the one which is more reproducible and in the combined data, so that people can just use this data directly for the data analysis. So this is the tab for, we did one-way ANOVA for our first experiment, and all the compound names and the p-values and the adjusted p-values that we give in the report. And we give the for the second same ANOVA, separate ANOVA that we did for the second experiment. So for the first experiment, this PCA plot shows that the LRT controls here, which are these blue dots, these how they separate from the rest of all these groups. So we don't see much of a training effect in these HRT samples, but we do see this LRT trained animals, how they become, how they are like different from these controls, and they like look more close to this LRT, HRT controlled and trained animals. And this heat map, again, for significant lipids shows the similar thing. These green ones are all the LRT controlled samples, and the blue ones are LRT trained samples. And these, so now in this cluster, you can see that they cluster pretty well with the high response to training rats after the training. And these low response to training controls separate from rest of the them. And again, two big clusters here that look a little different. So for the experiment, second experiment with LCR, HCR, and where we have three age uh, three age groups, basically the young, mature, and old animals. As you can see, the biggest separation here is on age. So these are all on PC1 axis towards the right side. These are all by young animals, both LCR, HCR, young animals. And then on this side are all the mature and old animals. So these ones right here are the old animals, and these ones over here. And this is a heat map for all the significant lipids that cluster really well on age. All the young animals, as you can see, all the young animals here cluster together, and the old animals are somewhere here. So you can see pretty big cluster of differences on age, and there's a lot of data to look at. So with the same plot, I just changed the legend in the next plot to see if just with these lipids, can we see how the line also is different in this. But we don't see with just these line differences, we have another set of lipids that change on line and age as well. And this this plot for these significant lipids shows that kind of variation we see only in the mature. These are, this is not, I'm sorry, this is not very clear, but these are, the black ones are HCR, mature animals, and the blue ones are LCR, mature animals. So we don't see much differences uh, in the LCR, HCR young animals and old animals, but we do see the LCR mature animals cluster come mostly in this cluster, which is, sorry, which is the old animals. So these LCRs cluster more closely to these old animals, and this one HCR mature animals cluster separately. So this is just an example of what, like, different way of visualizing the data. Plus, this is what we give. If there is an interesting pattern or visualization pattern, we do give this with the report. This is my last slide. So, any questions? Yeah. Uh, so I think there's a confusion with the terms here. So you're talking about standardizing or scaling versus normalization, right? So normalization would be just that we do metabolite by metabolite, uh, and then, uh, sorry, I'm saying the opposite. So standardization would be that we do metabolite by metabolite, and normalization would be that we do for the drifts and other corrections. So we do both, but we do at different steps. So standardization would be depending on what you're trying to do in your data. If you're trying to do, let's say, this kind of heat map, yeah, if you want to look at the correlation versus covariance, if you want, then you will do maybe a auto scale or any other kind of transformation. But so it depends what you're trying to do for standardization. But normalization you would do anyways. Yeah. Yeah. Yes. So that's the reason I combine these data sets, because most of the time people used to come back and say, how do I do this? What do I do with this data now? How do I pick which one I want to pick? So I think that's why I have combined it. But you are free to, like, look at it yourself. And if there is another way you want to look at these things, especially let's say if you want to check some of the compounds that are coming in both positive and negative mode. It's a good way to check also your data. Like, if you do it one analysis, something is coming out significant when you do it on positive mode data and versus the negative mode data. So yeah, if you want to look at it yourself more, you're free to do that. But I think the combined report, mostly we've taken care of all those issues, and you can just trust that data.